as Dean Davenport speaks, that nicely introduced Dr. Grant. Dr. Grant is our keynote speaker today, and we're talking about does the dye ever wash out? The emerging neuroscience of substance abuse recovery. Okay, thank you. Thank you.
Thank you. It is a great pleasure and honor to come here and speak, and particularly because they don't let me out much in terms of being able to speak to a community audience. Most of the time when I do get the opportunity to speak, it's at scientific conferences. And the topic I'm going to talk about, a little bit higher. Okay. The topic I'm going to talk about today, although we will dive deeply into particularly brain inhibiting research, I think has great value and practical implications for research and prevention, particularly recovery and treatment, as well as prevention. But before I get into science, I want to acknowledge my debt and gratitude to Dr. Edie London, who's in the audience, because I would not have done any of this without her support and her encouragement and intellectual collaboration. It just wouldn't have, none of this ever would have happened. So thank you, Edie. Okay, so the title of my talk is Does the Dye Ever Wash Out? The Emerging Neuroscience of Substance Abuse Recovery. It's emerging because what I'm going to show you today is primarily more provocative than compelling, and it's really more of changing the concepts of how we may approach substance abuse treatment and recovery. So most of you may have heard this over and over again, the NIDA disease paradigm, which is addiction is a chronic relapsing disease of the brain or dysfunction of the brain. This has been the NIDA motto for a good quarter century. But it raises an interesting question. And this question was made very explicit by David T. Courtwright in his article, The NIDA Disease Paradigm, where he points out that while this approach has been adopted and embraced fully by the research community, particularly the neuroscience research community, it is not penetrated to policy or treatment or community engagement with the problems of substance abuse. And one of the reasons that he points out of why that may be true is the implication from the definition that calling it a chronic relapsing disease, which is accurate. We know that addiction comes in cycles, and people have periods of abstinence, periods of treatment, periods of relapse. This is one of the major characteristics and problems with substance abuse. But by saying it's a chronic relapsing disease of the brain, like the sheet that has been dyed, the implication is the addicted brain could never become unaddicted. And you may recognize this as similar to the concept in 12-step programs, that one is always in recovery. One never fully recovers from addiction, but it's an ongoing process for the rest of the life. I won't get into the semantics of the difference between remission and recovery, but it may be something from the medical literature that we can fruitfully start thinking about what would be the difference between remission and recovery. But today I'm going to talk about brain differences in addiction, which is where most of the research has been. I'm going to go over that very, very quickly. Brain predictors of relapse, and then brain changes associated with recovery. And at the end, I will make quick mention of whether particular specific treatments change the brain, or what evidence we have of specific treatments changing the brain. And that's probably where the least data is available. Okay, these are the classic tenets that have underlined drug use research since, I don't know, maybe the mid-80s or so. Drugs are reinforcers. So that came out of the research on animal self-administration. Animals will press a lever to receive a drug. By definition, that makes it a reinforcer. And since they're reinforcers, they presumably have a hedonic component to them. So drugs are rewarding, they produce pleasure, and dopamine was identified as a brain signal for reward delivery. At least initially, that's what we thought dopamine was doing in the brain, the neurochemical dopamine. And further research found that dopamine is the final common pharmacological pathway for many drugs of abuse, if not all drugs of abuse. And if anybody has seen our director, Dr. Nora Volkow, and seen her talk, she presents a wide range of evidence suggesting that dopamine plays a central role in drug addiction. And you may have heard the more common sense statement that drugs hijack reward system. So, taking these as premises, you come to an approach for treatment that if you block dopamine, especially in a particular pathway in the brain, what we call the mesolimbic dopamine system, which involves the ventral striatum and the nucleus accumbens at the base of the brain, then you will block drug administration, and addiction will disappear on its own. So you block the ability of drugs to be a reinforcer. There's no reason to take the drug anymore, and so everything returns to normal. But that's an assumption. Is that really true? And it's turning out not to be true. It's not true in two ways. Drugs that block dopamine, various aspects of dopamine, have not yielded a therapeutic agent. And while we always hope that we will find an agent using this approach, we have failed to do it so far. And this is despite a massive effort to develop new drugs that target the dopamine system or target specific parts of the dopamine system. So when I read the David T. Kortwright's paper, I realized that maybe these assumptions are wrong. And maybe the assumption that if you block drug taking, maybe there are things that persist that will then lead to the vulnerability for relapse. So there's been an enormous amount of research on how drugs change the brain. And I'm not going to go over that. That's a whole lecture in of itself. Let me simply say that it extends from brain systems and circuits all the way down to genetics. And drugs have profound effects. And I could give you a laundry list of molecules, proteins, genetic expression, brain activity, brain cellular activity that changes during addiction. But we don't know whether or how long it takes for any of these changes to come back to a normal level. And this is critical because the assumption is that if you block drug taking, then the system will return to normal once the drug clears out of the system. This is the basis of detoxification. But all of you who have worked in the substance use field know that the process of recovery starts with the drug leaving the system, the person being clean and detoxified, but that is only the start of the process of recovery. So what happens once the drug leaves? What else is there? How deeply has the brain died? And this opens up the possibility that as we recognize that the brain remains plastic and modifiable throughout the lifespan, that perhaps the vulnerability to relapse after drug abstinence may be due to cellular adaptations that persist even in the absence of the drug. And these adaptations may be distant from the initial site of the drug action, both at the molecular level and may involve different neurochemicals, for example, glutamate, then dopamine, which is, or the mu opiate receptor, which is, or the nicotine receptor, which is this site of initial drug action. And the inference here is the adaptations persist in the absence of the drug being in the body or in the absence of signaling from the dopamine system. So blocking dopamine would be expected to stop the action of the drug, but not block addiction and not block the vulnerability to return to drug use after a period of abstinence. So the first question, and one of the easiest questions, nothing's easy, but one of the more straightforward questions to ask is, at a point early in abstinence, maybe after a day or two, is the brain in a state that you can use brain activity or brain structure, the changes in brain activity, brain structure, to predict who is most vulnerable to relapse? And while we may be quite a ways from employing brain imaging in the clinic because of the expense and because of the sophistication of the analysis that's needed, it still provides you with an insight that that successful recovery in abstinence may depend on the brain being in a particular state. Okay, so these studies, I'm mainly going to focus on human studies. Whether the brain state prior to abstinence or treatment may index the lack of a drug effect, or the absence of a drug effect, or resilience, the capability of the brain to be resilient, or maybe even the therapeutic concept of readiness to change. So one of the first studies in this area was done by Martin Paulus. I'm not going to go through the details of these studies pretty quick in very much detail, but what you see here is a list of brain regions. And the solid bars are people who, in a VA treatment program, who were followed for a year, followed at a year's point. And the groups were divided up into those who had relapsed and those who remained in abstinence. And the numbers on the x-axis represent brain activation during a decision-making task. I'm not going to go into the details of this task. The important point is there is a clear separation in these brain areas between those who relapsed and those who recovered. And this separation was evident within days of stopping the use of the drug. So some people had brain changes that made them vulnerable to relapse, and some people had brain changes that were predictive of continued abstinence. An area that has been of increasing focus, now let me go back to this for a moment. and mention that most of these areas are not the usual suspects in substance abuse research. You notice that we do not have the substantia nigra, the ventral tegmental area, which is where the dopamine neurons are located, or the projection areas into the striatum or ventral striatum, even though those are the areas where drugs of abuse act, particularly stimulants. But this is not specific to stimulants. I'm going to go across a number of drug classes. Here we see a study in where Insula activation predicted smoking relapse. Now you might note that on the top there, do I have a pointer? Is there one? Yes, no?
Yeah, it's just that.
Yeah, okay. So you'll notice this top area is the insula. This was not an area that was considered to be involved in drug use. It was really mysterious until, and I have to, this will be one of the sole mentions of my own research, the research I did with Dr. London, when we did our study of cocaine craving in the mid-90s. Surprisingly, this was an area that was activated when people viewed pictures of cocaine and was correlated with their cocaine response. This was quite surprising because nobody had ever linked the insula to drug abuse before. Since then, there have been multiple studies showing that the insula is important in mediating the effects of drug cues. You notice, though, in the relapsers, on the particular task that Dr. Paulus used, the insula decreased its activity, and it was the decrease in activity that predicted relapse. Whereas in the work that Dr. London and I did, and others have subsequently done across a range of drugs, when you present people with drug cues, the insula becomes hyperactive. And so it's not a matter of the insula being functional or non-functional, but rather it's dysregulated. It's not activated when it should be, and it's overactive when it shouldn't be. So this is work by Amy James at McLean Hospital, showing that smoking cues activate the insula right here, and that the degree of activation is predictive or correlated with whether the person subsequently relapses to smoking. It's not only individual brain regions that serve as biomarkers or predictive markers of relapse. One of the major advances in brain imaging research these days is the ability of development techniques that allow us to look at connections between the brain and crosstalk from one brain area to another. And this is generally just called connectivity analysis or network analysis. And in this study, it was connectivity between the inferior prefrontal cortex to one of the usual suspect areas here in ventral striatum that predicted relapse. So the less, that's the downward direction here, the less that those two regions were talking to each other, the less they were connected, the more likely was that the person would be relapsing. So relapsers had very low connectivity, whereas those who had normal connectivity remained abstinent. So again, this is a brain scan that was done after an initial detoxification, and then relapse was measured weeks or months later. And in fact, if you do a formal signal detection analysis, which is what you use for diagnostic purposes, as you go out, look at abstinence further and further out, the detection curve gets better and better, becomes more sensitive. For this particular circuit, this was work done by in Elliot Stein's lab at the NIDA program, between the medial prefrontal cortex and the temporal pole. And I want to point out that the temporal pole, while has a long history in neuropsychiatric disorders, was never a region thought to be very much involved in substance abuse. Yet the connectivity between those two regions seems to be a predictor of relapse. You may be wondering, why so many brain regions? Well, think of the brain as a community. And the community has a lot of different members, and those members form networks just like a community forms networks. And it is likely that these networks and these various brain regions may recover at different rates or may be associated with different phases of recovery. So personally, I am not concerned that we're not seeing the same brain regions over and over, although we are in some cases, for example, the Insula. But we're also seeing new brain regions that we had not previously considered. Okay, this is a study that I'm very excited about. And I should mention, unlike other speakers that you may have had in the past or you may hear this afternoon, I'm not talking about my own research. As a program officer at NIDA, I oversee the management of grant awards, like the are. So one of the great things of being a program is I get to view the entire field as my laboratory. And I get to promote research from a variety of sources. So I'm neither attached to these, I have no skin in the game, so to speak. But I get to see research at early stage, and this is one case where this is a project that's going to be conducted at New York City in opiate abusers. And I'm very excited about this because they're going to be taking a lot of measurements through a mobile phone app of opiate abusers who are maintained on methadone out of a methadone maintenance clinic. And then they're going to do brain scans about every couple weeks or so during the course of recovery. So we'll get a longitudinal, at the end of this project, we'll get a longitudinal time course for a number of brain regions during recovery. But what is particularly provocative about this study is being done by Paul Glimcher, who is a neuroscientist at NYU, and John Rotrosen, who is a treatment researcher in the area of substance abuse and one of the core members of the NIDA Clinical Trials Network. That they're going to use this mobile app to probe two important factors. One is risk-taking, the propensity to take risks, and the other is what's called delayed discounting, which is just simply saying, if I offered you $10 today and $100 in a month, which would you take? And it's been shown primarily through the work of Warren Bickel that drug abusers will take the $10 now. And this goes along with our idea that drug abuse is associated with impulsivity and a very short time horizon. They don't see very far into the future. So they're going to take those two measures along with other assessments on a regular basis, about every other day. And they're also going to note when these people have relapse incidents. So this is very provocative early data out of this study done by Anna Canova, who is a postdoctoral researcher with that group. On the left, and I'm sorry that it's fuzzy, this is the way the chart was provided to me, that the y-axis here, oops, at the y-axis and the blue line represents the discounting rate, and going up is worse. That's really the main message here. So as the discounting rate goes up, you tend to choose the immediate reward over the future reward, even though the future reward is much larger. And on the y-axis and in the red line, this is propensity to choose the risky option. And what you can see here is if we center the measurements on the point where the people relapse, way before the relapse event itself, in fact, about a week to 10 days, you can start to see a rise in both of these measures. They become more like, they tend to start choosing the risky option. They start choosing more of the sooner options. And then they relapse. And then those measures stay up for a while, for about a week to 10 days, and then they start dropping again. This is very provocative. This means that this could potentially be used at the community treatment level to identify a point in time where someone starts to be at risk of relapse. And because we take advantage of mobile phone technology, they could be messaged back, come back to the clinic. We think that you need some additional treatment. So this has a lot of promise, and we will get the brain measures to go along with this when they finish their project in 5 years. So hopefully in 5 years, I could come back and say, look at this wonderful research. We actually have a way of intervening with opioid abusers prior to their relapse event. Okay, the next question is, does the brain change with abstinence? This directly addresses the question I posed at first, does the dye wash out? And if so, how long does it take to wash out? The seminal work in this area was done by Dr. Volkow, where she showed that dopamine receptors, which is what you see here in color, these colored areas, these are PET scans where you have a radio tracer that binds to the dopamine D2 receptor. So the more red, the more binding of the radio tracer and the more dopamine receptors that are available. Normal controls, you see a healthy, bright red spot. In methamphetamine users, 1 month abstinent, you can see that the dopamine receptors are downregulated and are much lower. 14 months abstinent, they're coming back to normal. But that's 14 months later, that's a year later, before this component of the dopamine system has recovered. And how many treatment programs, or how many substance abusers stay consistently within treatment programs for a year? The implication of this, and the other data I'm going to show you, is we're going to have a much longer time frame in terms of supporting people in their recovery. And this brings us closer to the 12-step principle of that you're always in recovery. But I don't think that's true, that at least for the brain, that the brain is always in recovery. So we see evidence here after a year, there's recovery. This is more work from Dr. Volkow's lab. I'm going to go into it very much. It's just another indication that in this brain region, the thalamus, which is important in relaying information to the cerebral cortex, there's a degree of recovery. This is glucose metabolism in the thalamus, but in the striatum, the area that we associate with drug action the most, there's not much recovery, even within a year. Okay, brain structure. These are, this is not dopamine receptors, but that is rather this map is how dense gray matter is, where the cells are in the cerebral cortex. And this is work done by Mohammed Karev at Rita Goldstein's lab at Mount Sinai in New York City. And you can see that at a 6-month follow-up period, there is improvement in the gray matter levels between in a number of regions that are associated with the orbital frontal cortex. The inferior frontal cortex, which is over here, has been linked in inhibitory control, and I'll show you some more data with that. The ventromedial prefrontal cortex has been associated with decision making, and these critical areas show deficits in brain structure early in abstinence, but by 6 months, they're starting to come back. But it's 6 months. This is another study of brain structure. This was done by Hugh Garavan, showing that the amount of loss in this area, the anterior cingulate, which is another area that's going to come up over and over again, and the anterior cingulate has been linked to error detection and error monitoring and being able to correct behavior after errors. And of course, what is a diagnostic criteria of drug abuse? That you continue despite adverse consequences, you continue despite making mistakes and errors. Well, it looks like one of the brain areas important in mediating that behavior, the ability to detect areas, has structural deficiencies. And those structural deficiencies are correlated with the years of use of the drug, suggesting that some of this is pharmacologically driven, and it improves over weeks of abstinence. But note, we go out to 100 weeks here. That's 2 years to get a substantial recovery. But the good news is you start seeing recovery somewhere around 40 days. But again, that's 2 months. Here's another brain area. I mentioned the inferior frontal cortex and the insula. I keep losing my cursor here. That's over here. Same story. Full recovery is starting to look like it's going to take years. And remember that the insula is related to cue reactivity and craving, whereas the inferior frontal cortex is associated with inhibitory control. Okay, so this is going back to the work of Martin Paulus. Because of the initial results showing the insula involvement in abstinence, he has continued work looking at the function of the insula, and he's developed some very interesting paradigms for probing insula function. And in this case, he uses a brush, just a camel hair brush, to stroke along the inner forearm. And it turns out this activates the insula extremely well. It is not aversive. In fact, it's mildly pleasant. And what he finds is that the brain responds to this soft touch at baseline is attenuated relative to what normals have. They're not showing very much of a response at all, either to the anticipation of the touch or to the touch itself. But by 6 months abstinent, they're starting to show a hypo-reactivity to the anticipation of to the touch, and a hyperactivity to the actual application of the touch. And this is different from normal controls. Normal, healthy controls show an increase in activity to anticipation of the touch and less activity, relative activity, to the touch itself. So if you can predict something is going to happen, usually what we have found out over the years is the brain reacts in an anticipatory way, and if you can anticipate something, then you get most of your brain activation to the anticipation of the event and less to the event itself. The important point, though, is for this as a probe of the insula, 6 months before you start getting any kind of robust activity. So again, 6 months, what kind of treatment and support are we providing for drug abusers at the point of 6 months of abstinence? Okay. He has done a different type of study to probe the ability of probe the insula activity to an aversive event. And the insula has generally been associated with signaling aversive events. Discussed losses in a task where you have to make decisions and you lose or you make a mistake. This is what most people find with the insula. So he did a study using a restricted breathing probe, which means they just had a tube like you would use in anesthesiology, where he could vary the airflow. So one would have to exert more effort to bring in the necessary amount of oxygen. This is very aversive. Any of you have ever gone snorkeling or scuba diving and your airflow gets restricted, it induces a sense of panic. It's not fun at all. He didn't restrict airflow that much, but he did restrict it in a parametric way. So he had a condition with lower airflow and or lower restriction, and then a more restricted airflow. And what you see here is that there's not much activity to the anticipation, but across two groups, whether they are acutely abstinent or whether they are remitted, in this case 6 months to a year abstinent. And what you see is this response to the breathing restriction does not reach the control level of healthy controls until you get out to 6 months. So after a short period of abstinence, they're still not responding to this very profound physiological challenge to their health. 6 months later, it's more robust and is approached or equivalent to control levels. So again, this area that is the insula, that is reactive to cues and is predictive of relapse, does not regain its normal functioning until 6 months of abstinence. Same for the anterior cingulate, which I mentioned before, which is another area associated with the insula and the anterior cingulate are actually very heavily connected with each other, and they're part of what's called the salient system, the detection of important events in the environment, especially when they're unexpected or represent a threat to the organism. And you can see again that you do not see the normative pattern to the breathing load, the parametric breathing load challenge until 6 months of abstinence. And in fact, at 6 months, it's still not quite normal. What you see with the healthy controls. So they're hyperreactive in the insula. They're hyporeactive initially during acute abstinence. 6 months out, they become hyperreactive. to this aversive event. This may be a brain process that mediates the well-known clinical finding and animal findings that stressful events precipitate relapse. And it may be because the brain is hyperreactive to the hyperreactive in the anterior cingulate. And you find a similar pattern in you find a pattern slightly different in the inferior frontal cortex, which is important for essentially putting the brakes on for inhibitory control. And again, at 6 months, in acute abstinence, you have an abnormal pattern. They're very, very hyperreactive to a low amount of restriction in the airflow. They're hyporeactive to a high amount of restriction in the airflow, and then that starts approaching a normative level after 6 months of abstinence. Okay, here's a study that Hugh Garavan did on inhibitory control, actually probing for the inhibitory control function. And in this paradigm, you have to press the button when you see 2 X's in a row. So these letters are presented sequentially, and you have to press the button when 2 X's have occurred. And you see that there are some cases where the X appears alone. If you're lacking in inhibitory control, you'll make a commission error. You'll press you won't wait for the second X to show up. You just, oh, there's an X, bang, press the button. And what he shows is that in contrast to current users in the inferior frontal cortex, which you see in the area that's circled, that even though they didn't vary in the performance, the brain's substrates were different. And in fact, for current users, they show a fairly low level of activation. 3 weeks of abstinence, they're hyperreactive in this area. They have to get more brain activation in order to maintain a normative level of performance. And at 70 weeks, that elevation is still present. 70 weeks, about a year and a third, year and a quarter. They're still having problems with inhibitory control. No surprise that there's a high rate of relapse within the first year of abstinence because the brain has not recovered. The dye has not washed out. Here's another example where they measured not only the button pressing, but they also had to indicate if they thought they had made an error, if they recognized they had made an error. Again, you got the same pattern, that you start seeing hyperactivity at 3 weeks and 70 weeks. So initially, the brain is not recognizing that an error was made. It sounds an awful lot like the concept of denial. Well, maybe they're not lying when they say they don't know how much drug they use or they don't think they have a problem. Maybe the issue is there is a brain dysfunction and that impairs their ability to recognize that they've made a mistake or that there's a problem. And that brain function does not recover until maybe 3 weeks, maybe up to a year. Okay, this is another type of brain signal, and the only reason I'm presenting this is I'm not doing this to confuse you, but rather to show that this is a robust phenomenon. Brain changes during recovery are a robust phenomenon that we can see over a variety of different brain measures: brain structure, brain functional activation, and this is a neurochemical measure. And what you can see in methamphetamine users, this is work by Ruth Sallow, that short-term abstinent methamphetamine users still have depressed neurochemical signal in the anterior cingulate, and it doesn't return for 1 to 5 years. So again, it takes a long time for the brain for the dye to wash out, much longer than many treatment programs last. What? Okay, I won't go over this, but this is in smokers. It shows a similar pattern in that their response to an anticipated mistake changes from early abstinence to a later period of abstinence. In this case, it was following a treatment cessation program, a standard smoking cessation program, and this pattern initially shows a decreasing slope, but then at follow-up 3 months later, the pattern has reversed and they're shown an increasing slope. This is a little bit this paradigm is a little bit complex. I don't want to go into the details of it. Again, the important point is the pattern reverses, clearly reverses with abstinence. Okay, so to summarize so far, how am I doing on time? Okay, good. That we have evidence that the dopamine system, the anterior cingulate, the insula, parts of the prefrontal cortex do normalize, and these are systems associated with reward, salience, and executive control. That at the system level that we pick up with brain imaging, these areas recover, but it takes much longer than the standard 28-day treatment period. It takes much longer than simply having the drug wash out. There's something else that persists that takes a much longer time to wash out once the drug is out of the system. What other brain systems renormalize? Well, there's a multitude, as I showed you at the beginning, and we have not investigated this very deeply. So this is a direction for future research, both in humans and animals. What doesn't normalize? Or how much of these changes represent compensatory changes? So for those things that may not recover immediately, are there other brain systems that may be picking up the slack? And if there are, maybe we can target those systems or the functions they subserve as treatment targets. How long does renormalization take? Well, it evidently is taking weeks to months to years, and it may follow different time courses for different aspects of brain structure and function. And then finally, once we characterize these, the time course of recovery across a number of scales from molecular to brain systems, how can we accelerate it? Maybe these represent new treatment targets. So let me give you a provocative example of that concept. First of all, this is a very recently published paper out of a group in France where they had rats pressing a lever for cocaine, and then they put them back in their home cage and let them be abstinent for a period of time, 1 week or 4 weeks. And since rat lifespans are about 2 years, 4 weeks is a long time in a rat life. And the important point here is rats show a similar pattern, or at least the similar types of brain regions that we see in humans. So in particular, the insula, the nucleus accumbens, the anterior cingulate. Those areas show changes as abstinence continues. the rats are not doing anything when these brain images are taken. They're just generally resting. Also, subcortical regions like the amygdala, the hippocampus, and the dopamine system, they were able to pick up changes in the span of 1 week of abstinence to 4 weeks of abstinence. So, rodents may go through the same brain recovery process. If that's the case, we have good animal models to really dig into the mechanisms that may underlie the progress towards brain recovery. The best example of this is work first done by Yubin Shaham at the NIDA intramural program, showing in a task, in a paradigm where animals self-administer cocaine, then they're put back in their home cage. They have no opportunity to acquire the drug. They are abstinent for a period of time. In this case, they test them, they withdraw them for 45 days, right, so a month and a half. And what they find, counterintuitively, but consistent with clinical experience, is if you put them back into the Skinner box where they can press lever for cocaine, they press more avidly at 45 days of abstinence than they do on the first day of abstinence, right? So what does this say? That, again, if we think of the standard 28-day treatment period, yes, the drug is out of the system, but the vulnerability to relapse keeps increasing for a much longer time. Can we understand the mechanism that underlies this, what has been called incubation of drug seeking or incubation of craving? Yes, we can. Marina Wolf at the Rosalind Franklin School of Medicine in Chicago has developed or has investigated for a long time the AMPA receptor. The AMPA receptor is a receptor that binds the neurotransmitter glutamate, and it mediates the straightforward excitatory effects of glutamate. Glutamate has multiple actions, and there are quite a few different types of receptors, but this is the classic excitatory neurotransmitter receptor. And what she's found is AMPA receptors increase in density in the nucleus accumbens over that 45-day period. And further work that she's done has found that the AMPA receptors that increase are actually an unusual type of AMPA receptor. They're not the usual AMPA receptor, they're a variant. They lack a molecular component called the glutamatic receptor 2 subunit. What's important about that is it's not the usual type of AMPA receptor, and it doesn't have the functional characteristics of the usual AMPA receptor. It emerges late in abstinence, and it allows calcium ions to come in to the cell. And calcium ion entry is not only excitatory at the electrophysiological level, but it is also an important signaler for biochemical actions and can activate various enzymes that are important in membrane structure and even reach all the way into the genome and change gene expression. And what she found with Yubin Shaham is, yes, these receptors go up. Sorry. The receptors go, these unusual AMPA receptors go up, but if you block them, unfortunately, the drug she used to block them is a neurotoxin and it can't be given systemically. But if you block them, then you don't see this incubation of craving. So if you can stop these AMPA receptors, these atypical AMPA receptors from functioning, then you don't get the incubation and craving, you don't get the vulnerability till relapse. You have a new treatment target. It has nothing to do with the action of the abused drug itself, but rather it acts on an adaptation the nervous system has to initial exposure of the drug and then subsequent withdrawal. So here's the model that when you have cocaine abstinence, you start seeing these receptors being expressed. Glutamate's action, what it does as a neurotransmitter, changes because the receptors are different. And those atypical receptors increase during abstinence, and it is that cues that can precipitate reinstatement of drug taking in rats may be mediated through the medial prefrontal cortex, which is equivalent to the anterior cingulate that I showed you with the human brain images. And that leads to vulnerability for relapse. And it turns out that this is an excellent review, number one. And it turns out that there is a different entry point for manipulating these atypical AMPA receptors. And that's through a different glutamate receptor called the metabolic glutamate receptor 1. And activation of the metabolic glutamate receptor 1 normally suppresses the expression of these receptors. So by activating the glutamate receptor, metabolic glutamate receptor 1, and GLUR1, you can suppress these atypical AMPA receptors. There is also a link in the biochemical process with endocannabinoids. So the cannabinoid receptor system is involved somehow, and that's a topic for future research. We also have, this was done obviously with stimulants. This needs to be replicated in other drugs that we've used like nicotine or opiates, but at least we have a handle and empirical evidence that there are ongoing changes at the molecular level that occur that take a while to appear after abstinence and increase the vulnerability for relapse. Okay, final set of results. Can treatment change the brain? We have very little evidence on this, and I'm only going to emphasize one part of this for the sake of time, but it's a very striking finding, very unexpected. Okay, these are PET scans again, and the color, again, is red is more binding of the radiotracer of the PET scan to dopamine receptors, in this case, again, the D2 receptors, but it's a slightly different paradigm in that you give a drug, methylphenidate, which acts to release dopamine and to increase the synaptic concentration of dopamine, very similar to cocaine. And after you give methylphenidate, you get a reduction in binding because the endogenous dopamine is kicking or bumping the radiotracer off the receptor. Right? So more dopamine is less red in this case, right? And it's a good thing. You want it to be less red. And what she did is her initial application was to see whether a standardized 12-week community management, contingency management with community reinforcement, a standardized manualized treatment program that is shown to be efficacious, will change the dopamine system. will the dopamine system recover? Now remember, the prior slide I showed you took a year for the dopamine receptor to recover. But if you give this treatment program, will the dopamine receptors recover faster? And what she found was counterintuitive and against her hypothesis. The dopamine receptors did not improve with treatment. There was no change, and I'll show you the quantitation, between baseline prior to treatment and after the 12-week treatment period. Well, that's disappointing. But when she did a post hoc analysis of those who remained abstinent, as measured by the vouchers they got, those who remained abstinent, it turns out that those who responded to treatment and provided clean urines had better dopamine to begin with prior to treatment. They did not show the reduction in dopamine receptor function that you see with that we thought was characteristic of all cocaine abusers. And by the way, there was no difference in their drug abuse history or how their time of last use prior to entering treatment or the other types of clinical measures. So this top, I keep losing my cursor. This top is up here, where it says responders, that's the normative pattern. That's what you see in healthy controls when you give methylphenidate as a challenge. On the bottom, you see the non-responders, and you see that they have very low dopamine receptor tracer binding to begin with, and it's not reduced after methylphenidate, which means dopamine is not being released. So they are hypodopaminergic, as we always thought. The surprise is that the state of the dopamine system prior to treatment determines whether they will respond to treatment. And here's the quantification, and you can see it's a quite striking difference. There's no difference, very little difference between responders and non-responders in terms of baseline dopamine receptor availability, but there's a big difference when you give methylphenidate and you look to see whether dopamine is released by methylphenidate, and it's not in the non-responders, whereas responders show a more or less normative pattern. And you can see that the non-responders, by definition, show a lot of cocaine dirty urines compared to the responders, and the responders earn a lot more vouchers for the contingency management than the non-responders. So treatment in this case doesn't change the dopamine system, rather dopamine determines the response to treatment. If we had a way of easily getting these measures that did not require the time, expense, and sophisticated equipment needed for PET measures, we would be able to say at entry into treatment, your brain is in a state where contingency management will work, has a high likelihood of working. And you have a brain where contingency management is unlikely to work. So just like with an infection, we can triage patients to a treatment that would be effective. The trick is going to be finding another measure that we can use as a predictor of treatment responsiveness. I'll skip over this. This is just showing that the brain changes with a standardized cognitive behavioral therapy and contingency management. So limitations, these are provocative, not compelling evidence. We have a long way to go. Some of these regions have been implicated in other psychiatric disorders, depression, anxiety, schizophrenia. So these may not be specific to substance abuse, but rather they may be what's called pseudo-specific because they cut across a number of disorders, or maybe we're tapping into a system that's integral to providing behavioral change. And all these studies are correlational, not causal, so we've got a ways to go here, but this represents a research agenda for the future. I'm not going to summarize this again, but this leads to the concept that the way into addiction, that is the action of drugs, the pharmacological action of drugs, is not necessarily the way out. That is, blocking the drug or detoxifying and getting the drug out of the system may be necessary, but not sufficient for recovery. So this is a chart of Dante's Inferno, where you enter and you can't go back out again. You have to go all the way through to the deepest depths of hell. So I've said too much, haven't said enough. Thank you for your attention.
Questions, but I think your Dante slide also brought to me, you got to stop people from using drugs in the first place.
You do?
I think that's the lesson I learned. Is once you're on it, it's so hard to get off and change your brain. We've got to work on stopping it in the first place.
Yes.
Two more questions, Norma and Deborah. And microphones.
Thank you. Hello. Thank you for the information on that. It's so palatable for us who are not neuroscientists. Thank you very much for that. We have a people back here. But you might have such a wealth of a collection of the data from the various research projects that funded. How does this translate to policy? How does NIDA get this information to the legislature? And I'm asking here on the county because we're having some major transformation with our contracted provider system. And things that may be going to Medi-Cal, which doesn't take on a whole lot of things, and shortening the treatment as opposed to expanding it. And we have to acknowledge that basic knowledge already that we do need much more time. How does that translate to policy?
So everything I've shown you, except for the studies of predicting opiate relapse, are published studies in scientific journals that you can access, you can find through PubMed and access through normal laboratory, normal academic resources. And maybe the first step is just to print them all out, put them in a binder, and slap them down on the desk and say, look at this. Here's scientific empirical evidence that the brain is not normal after whatever period of treatment. You're trying to reduce the treatment period, and here's evidence that the brain is abnormal, and you're setting people up to fail by not providing extended treatment.
And I think for Manal might be addressing some of those issues in her talk, maybe. And I think part of our theme today is to try to bridge drug addiction from the beginning to the end, including trying to change policy and changing behavior we do in the community.
So let me add my thanks for your presentation and ask a two-part question. The first is about how far the field has come, and that's just based as a former clinician who did physicals for methadone maintenance clinic back 40 years ago in Boston. I was struck by how the research and the clinical aspects of substance abuse treatment were separated, and that there was almost a dichotomy, and the treatment was criminal. It was not medical. What I did was based on some legal criminal formula that was mandated. So while in clinic I might be able to adjust insulin or adjust the medication based on an individual patient, when I was in the methadone clinic, I had no clinical responsibility other than this checklist.
Mm-hmm.
That dichotomy always bothered me, and I'm wondering how far we've come. And then specifically your presentation at the end with the question of how can we predict the responders versus the non-responders. I'm wondering if we do have enough clinical ability and history to really perhaps predict that someone who knew how many times somebody has been in treatment and not responded, and the different drugs. So sort of related, but your thoughts about that.
So, addressing your first point, thank you very much for mentioning the advances we've had. That means I did my job. That's what I set out to do when I joined Dr. London's lab, and then went down to headquarters and started a portfolio in cognitive neuroscience. So, yay, I'm done. Right, my assignment's over. But obviously there's a lot to do, because these are generally small studies. We need large-scale studies. We need really rigorous longitudinal studies, like this being done on the onset side through the adolescent brain cognitive development study, where we're tracking vulnerability to starting drugs. We need to do that on the recovery side as well. To your second point, Dinah Martinez actually did a follow-up with exactly answering the question. Turned out that the dopamine signal, or lack of dopamine signal, predicted 70 to 80% of the variance. And adding the standard clinical measures of drug use frequency and other some things added a little bit more and got the prediction up to maybe 89, 91%. But it was the dopamine signal that was far and away driving the prediction. And that's published.
Thank you so much. Great. Thank you. Microphone. I'm requiring people in the back to come up. We're way too crowded in the back, way too empty in the front. So please come up to the front before the next speaker.
While you're doing that, could you please fill out this form? Ms. Morris in the back, Tamika Morris, will be collecting these, or put at the table at the break. We